All right, thanks everyone for joining. If you haven't already in the room, feel free to grab a donut, grab some coffee. You're welcome to it. My name's Claire White. I'll just give a brief overview before I hand it off to my colleagues. I'm your local account manager. I'm based here in Chicago. For Illumina, I cover all of University of Chicago and the academic and industry accounts in the Chicagoland area. I'm joined with Logan Silber over here, who's our sequencing specialist. He'll be giving the first part of our presentation, as well as Chevelle Blackburn, who will give the second part of our presentation, kind of a more technical overview of kind of how to run our NextSeq system. Just wanted to do a brief summary of the two cores on campus so you guys can put in your mind kind of when we talk about these two instruments, we're mainly going to be focusing on the NovaSeq X Plus, which is our latest high throughput system that's available at the University of Chicago Genomics facility. Peter Faberson in the back, who runs that facility. They actually just celebrated their 200th run. So they've been doing a ton of sequencing on the NovaSeq X. The NovaSeq X already utilizes XLEAP chemistry, which we're going to focus on today. So that's more of a service from sample to data, kind of send either your samples for library prep or even your finished libraries for sequencing, kind of get back your data. They also have access to our MySeq system, as well as our NextSeq500, as well as microarray capabilities. So if you're interested in doing genotyping or methylation type studies, you can do that at that facility. The other core I want to mention is our single cell immunophenotyping core with Fatih helps run. This is more of a walk-up service. You sign up, you get trained. It's open 7/24. So, if you need a quick turnaround or you just want some more hands-on experience, they specialize in single cell as well as spatial, and they have access to a MySeq and an XSEQ2000. With an XSEQ2000, we're just starting to release XLEAP-SBS kits on it, which is why kind of that's part of this conversation today too. Well, these slides will be available to you, and I have links to both the cores if you guys need to learn more. All right, with that, I will hand it off to Logan.
Good morning, everyone. So my name is Logan Silber. I am your sequencing specialist with Illumina. So that means my role is I assist in consulting you guys across the whole NGS workflow. So thinking library prep, sequencing, data analysis. If you guys have questions about your experiment and kind of the best approach to getting to that insight you're looking for, that's my role here at Illumina to assist you to get to that goal that you're looking for. So with that, I want to kick off with first a question to see who here is familiar with Illumina's SBS or sequencing by synthesis chemistry. I see a few hands back there. So for those not familiar, our sequencing by synthesis chemistry is just our kind of general sequencing chemistry that's used across all of our platforms for reading bases based on the libraries that are loaded across our instruments here. So when we're talking about XLEAP-SBS chemistry, this is just our newest iteration of this process here. It's the exact same chemistry and workflow and how we incorporate bases and read them and have the instrument, put out the data on it. But what we've done here is we've modified every component of that SBS chemistry, every polymerase, every X blocker, every X linker, all the dyes to be more stable across the board. So this allows us to even actually ship XLEAP reagents at room temperature. So they can be shipped lyophilized. And then, for example, on the XLEAP specifically here, these can be then resuspended on instrument. So this allows us to actually be more sustainable when we think about our sequencing projects in general. But we're going to be talking about how this chemistry that we've modified and created allows for better quality reads, faster run times, and then also reduces the cost of your sequencing projects too. So currently right now, there are only two instruments where this is this chemistry is being used, the XLEAP version specifically. The first is the NextSeq1000-2000 systems here. So for those who have used the single cell core, might have used the walk-up service, just a little bit of a recap on some of the attributes of the system. This is a dry instrument, which means that you don't have to worry about maintenance washes. All the reactions take place on board the instrument. So you don't have to worry about denaturation off instrument. You can just take your library, load it directly into the cartridge, click start, walk away. It's an extremely easy to use process for a mid-throughput instrument here. Additionally, we have our Dragon secondary analysis pipelines on board this instrument. So say that you have your FASTQ files, but you want to just take them to BCF file right away. We have pipelines on board for RNA-seq applications, single cell applications, germline and somatic applications, enrichment and amplicon-based applications. You can get to the reads that you want all for free off the instrument after you click start too. So any questions about that, we're also always happy to answer that too. Additionally, all these advancements, of course, are powered by XLEAP as well, or some of these kind of things I'll be talking about. But This is our second newest platform in general, and advancements to hardware, including being able to reduce the pitch of our flow cells. So fitting over 1 billion reads in some very small areas here has allowed us to kind of get this throughput and cost effectiveness that we're going to be talking about too. Additionally, for those who might not have used the core here for using for high throughput studies, we have the NovaSeq X Plus on campus here. So we have, as I called out, those extremely high-pitched flow cells, which means that you can fit more unique libraries or individual libraries onto the flow cell. We have we've advanced our base calling algorithms, which just means here that because of how fast we're going to be moving with the sequencing, we've improved our RTA, or real-time analysis software, to be able to read and generate data on instrument as fast as we're doing here. And that also kind of comes into play with more hardware advancements, including improvements to optical cameras too. Additionally, that Dragon software that I've just mentioned, the NextSeq2000, it's only available on the 2000 as well as the NovaSeq X. So if you have applications like whole genome sequencing, as I just called out, you can get that BCF file all off instrument, no charge to analysis cost there too. So saving use costs throughout the entire library prep process there. So we've kind of talked about some of the instruments that use this technology, but we're going to get a little bit more into the weeds of kind of what this means exactly, and kind of what flow cell configurations are available to use this, and how it compares to our standard SBS chemistry here. So to start, as you guys have heard, and as Claire has just called out, we've just came out this XLEAP chemistry on the 2000 as of this year. This is actually in the form of a P4 flow cell. So for those who have maybe used the walk-up core, there are a few different flow cell configurations: a P1, which offers 100 million reads per flow cell, a P2, which offers 400 million reads per flow cell, a P3, which offers 1.2 billion reads per flow cell, and a P4, which is now at 1.8 billion reads per flow cell. This XLEAP chemistry is also going to be coming to these current iterations, the P1, P2, P3, and these flow cell configurations that will use XLEAP, you'll see a big reduction in cost when using the XLEAP versions of these flow cells later on this year. Definitely in around June, this should be coming out here too. So we'll talk a little bit more about pricing in a second. So when we think about also run times and kind of data output here, what the best flow cell configurations are for your experiments, there are going to be 4 different sort of outputs with that 1.8 BU flow cell here. We have a 50 cycle kit. This is good for kind of smaller RNA-seq experiments for gene expression, single cell, spatial, well, maybe not single cell depending on which approach you're doing, but spatial profiling here. The 100 cycle kit, which definitely falls a little bit more into the single cell sequencing for many of the different single cell library prep providers out there. 200 cycle kits, which fits very nicely for enrichment-based approaches such as exome sequencing or doing larger panels. And then we have the 300 cycle kit, which you start to get into kind of whole genome sequencing. If you have a trio or maybe 4, maybe you're trying to do a lower coverage approach for doing whole genomes, this is kind of where we can fit in, because we're approaching that lower end of the NovaSeq X in throughput here. So this is really a great instrument for speed. And if you have your kind of smaller projects that you're looking to get started on, get turnaround time on, you know, having access to the core and be able to walk up, this is going to be a great application for you guys here. So when we kind of compare that in context for maybe you haven't used a P3 flow cell, or maybe you haven't used some of these flow cells, just want to show this slide to show how it compares exactly here. You know, one thing with Illumina is that we have our specs that we publish, but people generally tend to see that they get a lot more than they generally would expect from the specifications of our flow cells. And this is something that we see is consistent too with the P4 flow cell as well. You know, for the P3 here, you know, our specs call you'll get about 360 gigabases per run. You know, in this kind of example here, with some internal testing with a PHYX library at 411 gigabases. For the P4 flow cell with that 1.8 billion compared to the 1.2 billion, you know, you're expecting the 540 gigabases, but from our runs, we've been seeing that you can get, you know, here at least 639 gigabases. Also, with this output and data, you're seeing that there's actually a reduction in time for your run. So instead of seeing, you know, having an increase in runtime to get this more data, you're actually seeing, you know, 4 hours less here compared to the P3 flow cell with that standard SBS chemistry. Additionally, we're going for our specs, we'll be saying that our Q30 scores, or the errors that we see in 1 out of 1,000 bases, at least 90% or greater of the runs will be, you know, have a Q30 score of 90% there. And so you can see we're calling 90, but we're seeing 95.63%. We're going to get a little bit more into the weeds and show you exactly how many reads are in Q30 or even Q40 with this XLEAP chemistry. And just at the bottom here, just kind of showcases the average of how many runs we tested between the P3 and this P4 flow cell too. So let's kind of compare some of the chemistries that we're going to be seeing coming out later this year, as well as how the P4 compares to some more on-market work here. As I called out, we're seeing similar overperformance compared to the P3 flow cell. So this here, we're using a P2 SBS chemistry, the current SBS chemistry. And then the orange here is the P2 XLEAP chemistry. So similar overperformance that we're seeing, as we called out before, that you get with Illumina reagents. But you're also seeing a reduction in error rate for that first read, 33% here, and then up to 41% error reduction for read 2. Additionally, you have that reduction in runtime that I've called out before, going from 29 hours to 23 hours with this P2 flow cell. So a one-day run that you load it, say you load on a Monday afternoon, Tuesday afternoon, the run will be done. And if you want to have analysis kicked off, you can have your VCF file by the end of the Tuesday, by that point. If we're going to compare this to the P4 flow cell to the P3 flow cell, we see that here that we used our TrueSeq PCR-free libraries. So this is generally used for kind of whole genome sequencing approaches. We see that once again, this makes sense, this huge kind of difference in output. This makes sense based on the specs that I just showed on the last page. But from a P4 to a P3, you're getting a 52% more output. And then also you still see that error reduction even on these higher output flow cells. So 37%, 33% for read 1 and read 2. And then also still that reduction in runtime. And as you'll call out and see with some of our list pricing when we're talking about XLEAP chemistry, the XLEAP P4 flow cell will actually cost less than the standard P3 flow cell too. So all these benefits that you're getting for running on your own, you'll see, and you won't have to pay more to get these kind of benefits too. So as we kind of keep looking at some of these results and how XLEAP compares to some of this current on-market standard SBS chemistry, we still see that once again, P2 or the P4 compared to the P3 for a exome library specifically, depending on how many people are doing large enrichment panels or panels in general, we still see great output and great results here across the board. We're getting a 16% higher output when we talk about our specs, as I've shown before, and also 48% more output when compared to the P3 again. So this means that you can get more exomes onto your flow cell in that case. And so when we think about some of these projects, the savings you see with sequencing, you can kind of convert that into doing more libraries for your sequencing run to get more data, more results to help strengthen your hypothesis and your paper that you're working on. So additionally too, you still see, I'm sure you guys are getting the picture by this point, that the XLEAP is providing better, or lower error rates across the board when compared to the standard SBS. Read 1, read 2, 33% better, 31% better. And then when we talk about relative cost per sample, you can see when we think about the P3 flow cell with for exome libraries here, you can see this decrease, and you're even approaching that lower end of the NovaSeq X with that 1.5B flow cell. So say you need a fast turnaround time on your samples, you can go walk up, load this library. But of course, as you think about your projects and as you scale, you're going to get the best price per gigabase as you get to the NovaSeq X. Just the NextSeq2000 is good for kind of starting off some of these pilot projects you might be doing and getting that for a fast turnaround there. So as we think about even longer reads, so maybe you're doing 16S sequencing, or maybe you're doing, you've generated your own amplicons that need quite a bit of read length here to get good results. This is how our XLEAP SBS chemistry compares for the 600 cycle kits on our current P1 and P2 flow cells, and then when they're powered by XLEAP as well. So same sort of theme that we're seeing. There's a error reduction here when we're using our XLEAP chemistry, up to 43% with this read 1. And about 49%, close to 50% here when we're talking read 2. Additionally, that we see that across the board, as you get to those end runs, for those who might have done 16S sequencing or kind of longer amplicon sequencing on the MySeq, you normally see the error rate start to increase as you got to the end of your runs with the read 2 especially. And we made some improvements with that with the standard SBS chemistry on the NextSeq2000, but you see we've even taken that further with this XLEAP SBS chemistry well into the 600th cycle of our sequencing run here too. So a lot of benefits here to using this XLEAP chemistry and providing stable results across the board for your applications that require these longer read lengths. Additionally, when we think about Q30 scores, we kind of talk about those and use those as our specs generally. But when we unbin our Q30 scores and just look to see what it looks like across the board from Q30 to Q35 to Q40 to Q45, we see a large majority of our libraries at the end of the day end up actually in that Q40 bin when looking at the NextSeq2000 here too. So you can be certain that the reads that you're seeing, the likelihood of an error is 1 in 10,000 there too. So very stable and very high quality reads that you're getting with the NextSeq2000 XLEAP chemistry across the board. So we're going to shift gears a little bit and talk about XLEAP chemistry on the NovaSeq X, and kind of put that in perspective about how this compares to the previous NovaSeq instrument, the NovaSeq 6000. We can kind of use this high-level overview to showcase how XLEAP is powering the NovaSeq X. With the NovaSeq 6000, if you wanted to use a flow cell that used 10 billion reads of flow cell, it would take you about 48 hours to run that, about 2 days to get that throughput. And you get about 24 whole genomes on that 10B flow cell, just for reference there. With the NovaSeq X, that same output, a 10B flow cell, you can run that same amount in 24 to 25 hours there. So pretty much 2 times faster in this sort of approach. You still get those 24 samples and you're getting data a lot faster. Additionally, with XLEAP, as well as some of the improvements to the NovaSeq X, we have even higher throughput flow cells on the NovaSeq X in the form of the 25B flow cell. So you can fit up to about 64 whole human genomes for reference, or about 1 million cells if you're doing single cells on that 25B flow cell too. So a lot more room for scalability and throughput on the NovaSeq X as you move with your projects across the board. And you have access to both these instruments, as I called out, between the single cell core and the UChicago core as well too. So a lot of room to grow here. And then when we think about how this XLEAP chemistry compares for the NovaSeq X, I'm sure as you guys are figuring out, as I called out, you'll see that still see those improvements with XLEAP on the 6 or on the X as well. We see a reduction in percent phasing with the XLEAP chemistry, as well as a reduction in error rate. And then we also see an increase in incorporation time, up to 3 times faster relative to that standard SBS chemistry on the NovaSeq 6000. So calling out once again, if you're loading this in the NovaSeq X, you're going to see that improvement to quality scores when we think about binning. And this NovaSeq X 10B run, we see a yield of about 3 terabases of data here. But when we unbin those Q scores, we can see that a large majority, about 2.41 terabases, are actually above that Q40. So the reads that you are getting from the core, from the NovaSeq X, they're going to be high quality with what you're getting here too. And just to call it, this was used, of course, with our human PCR-free whole genome sequencing library preps here. So 2x150 run type. So when we kind of take a look at this piece by the different flow cell configurations specifically, and seeing how, for example, 1.5B flow cell compares and what the expectations are, we can see a few things here. So here we're looking at different library prep types and the kind of amount of total yield for each of them. We've done quite a bit of internal testing with our library preps to ensure that what we tried out, you can guarantee that these libraries will work on a NovaSeq X here. We've tried out our TrueSeq DNA-free library, our Illumina DNA PCR-free library. And then if you're also interested in whole genome methylation sequencing, for example, we've also tested out a few Zymo kits for bisulfite sequencing. So we know that with XLEAP, you can still get good results with some of these approaches here too. Additionally, we've also tried out a variety of enrichment approaches in the NovaSeq X. So we have our Illumina DNA exome, where you can get full exome coverage. And then we've also tried another enrichment kit with the twist as well here too. And then, of course, we'll talk a little bit more about different variety and different scales of experiments we do with RNA-seq specifically, but we've also tried out our total RNA prep on this kit as well. We're getting great yield here too. So if you're interested in non-coding and coding regions of the transcriptome, we have a kit available for that, and you can get great yield from it too. So when we talk about the Q30 scores, once again, we can see here that for our spec, we're calling out about Q30 of about 90%, as I showed from the flow cell configurations or from the spec specifically. And we can see that we're well above that here. We're in the 95 range for the PCR-free. We're about 90 for Zymo-seq or the whole genome bisulfite sequencing. And for those who might have done methylation sequencing or bisulfite, this is pretty stressful on the DNA that you're going to be working with. So you can still see we're getting good results from the sequencing run itself here too. And that also is the same too when we look at the Q scores for the read 2 here. Additionally, we can take a look and see the Q30 scores remain stable for both enrichment applications as well as that total RNA prep approach as well. So no matter what application you're working on, you'll see these Q30 scores remain stable with a specific type of flow cell configuration. Now, when we move over to the 10B and 25B flow cell, so a lot more output that we're talking about here, you still see stability and high quality scores across the board, even as these run times go on a little bit longer here as well. You can see here that our yield, we've tested out once again in a few different libraries on the 10B and the 25B. And you can see that yield is exactly as expected, remaining above our specifications. And then also to the quality scores remain well above what we would expect from our specifications too for each of these applications. And there's an application that you're curious about and you might not see in some of these slides that we were sharing from internal testing, please feel free to come to me and or ask any questions about them too. Happy to share what we've seen internally if we have any data as well. But once again, also calling out that you will see quite a bit of your quality scores being in the Q40 range too, with the 10B flow cell as shown here. And so we're talking about these specific specs here, but just some kind of maybe a teaser, for example, we're seeing with the XLEAP chemistry that we can get pretty far out, as I called out that 600 cycle kit, showing even potential to possibly go even further than 300 cycles in a specific read when we think about read 1 and read 2. So a lot of potential for XLEAP to still grow, and as applications evolve, if the need for longer reads, or at least longer amplicon sequences to be generated on these platforms, I think that's something that Illumina is very much taking a look at. And if you have any questions or if there's a longer read, 2x500 or something like that you're excited about, please let us know because we can always pass along that feedback as well. So when we talk about cost now, for a P4 300 cycle kit, we're looking at about $9 a gigabase compared to for list price here. So when we think about what that means, when we compare like a P3 or a P2 or P1 flow cell to their XLEAP configurations here, you're going to see about a 30% reduction in price from P3 to P3 XLEAP, P2 to P2 XLEAP about 25%, and P1 to P1 XLEAP about 20%. And this does not, there's nothing specific here that you need to do to get these XLEAP chemistries. This is a free software upgrade, so this will be coming to you very soon with the P1, P2, P3 configurations, but already the P4 is more cost effective than the P3 right now. So something to look forward to, and as you plan out your future experiments, and if there's more information you want about list pricing, don't hesitate to come to us afterwards as well too. When we think about kind of relative cost per sample and how it compares from a P3 flow cell to a P4 to a 1.5B, you can see that P4 flow cell is approaching that NovaSeq X 1.5B sort of flow cell price per sample. Whole exome sequencing, you're seeing that it's very close to the 1.5B, and there's quite a reduction, about 40% here from P3 to P4. Transcriptomics, you're getting a lot more samples on with that P4 flow cell, so that price per sample is decreasing. That 1.5B pricing is still going to be a bit better here, but you're getting close, and so speed and turnaround time is important for your project. As I called out, you have access to this here. And then for single cell, you can see that it's still going to be pretty close to that 1.5B here at the end of the day. So I just want to turn over quickly to the NovaSeq X pricing. This is just generally for kind of price per gigabase, price per million reads for our list pricing specifically. You saw that with the NovaSeq, the NextSeq2000 for a P4 flow cell, it was about $9 per gigabase, and then for that 300 cycle kit. So you can see here, the gray bars are the NovaSeq 6000 that use our kind of standard our standard SBS chemistry. Well, the NovaSeq X uses the XLEAP chemistry here. And you can see these kind of same comparisons, that S1 flow cell compared to the 1.5B, the S4 to the 10B. You're seeing a huge price reduction just from S1 to 1.5B already, $5.20 per gigabase, 10B, $3.20, and then of course, 25B, $2 per gigabase there. So we're looking at about a $250 whole genome at the moment with the NovaSeq X2. So this is kind of the platform for these larger projects if you're thinking kind of, these larger scale experiments. And just to put this in context, when we think about millions of reads too, just because I know there's a lot of interest with single cell experiments, RNA-seq, you can still see that we're getting under $1 for a million reads on that 10B flow cell and 25B flow cell. So the 2000, great for that speed and flexibility, kind of getting started with your experiments. And as you kind of build out what you want to do, this is where you can start to scale more, all available for you on the University of Chicago campus. So just talking a little bit about flexibility and applications a little bit further here. So the NextSeq2000, there's going to be about 14 different kit configurations with 4 flow cell options. So as I called out, the P1, P2, P3, P4, we'll have an 100 cycle to 600 cycle kit on that P1. So great for everything from spatial to 16S sequencing, all the way up to that 1.8 billion reads for larger single cell and spatial projects, whole exome sequencing, even doing a few trios too. So going to be a lot of flexibility depending on what your experiment is. There's probably a flow cell configuration ready for you for your sample size that you might have for your project. Putting that into further context of which applications fit better, we can see that there's quite a bit here, whether it's, whole genome sequencing and liquid biopsy, that's going to be where kind of P3 and P4 fits a little bit better. So maybe you're looking at, cell-free RNA, maybe you're looking at different biomarkers, liquid biopsy sample. The 2000 still operate pretty well there. You still have your single cell and spatial profiling and your RNA-seq. So great for counting applications across the board, depending on how many samples you have. Repertoire profiling, which needs that 600 cycle kit to get the VDJ regions of the antibody chain, the chains that you are interested in here specifically. And then, of course, you have more for like targeted metagenomic sort of applications as well. So a lot of applications that you'll be covered by. And we kind of put that into some of perspective with some of the more popular applications. For a 40x genome, you can get about less than 2 days with that P4 300 cycle kit. You can do, if you're focused on a specific amount of pathogens, maybe in a sample, maybe you're doing like a wastewater sequencing sort of approach, and you're looking for, kind of pathogens out and seeing what's currently out in the ecosystem right now. You can get that done in about an 8-hour shift with a 1P 100 cycle kit with XLEAP. Additionally, small targeted panels and single cell, that P400 cycle kit, P2 300 cycle kit can be done very fast here as well. So really increasing turnaround time if you need an answer fast in these cases here. I know there's a lot of interest in RNA or RNA-seq or counting applications such as single cell and spatial too. So I do want to take some time to really hone in on what this means for you, how many samples you can fit onto each of these specific flow cells. So whether you're doing the 100 million read flow cell or that 1.8 billion read flow cell. So kind of taking a step back here, we're thinking about a P4 flow cell and how many samples you can fit on. If you're only focused on the coding regions, so anything with a poly A tail, you need about 25 million reads per sample in those sort of cases. So you can see here with a P3 flow cell, you're looking at about 48 samples, and this then can jump up to 72 samples of the P4 flow cell. Once again, calling out that the price for this flow cell is less than the price for this flow cell. So you can use the savings from the sequencing run to spend on more library preps to expand your project further, as I just called out. Additionally, if we're focused on the whole transcriptome, coding regions, non-coding regions, generally we recommend about 50 million reads per sample. So for a P3 flow cell, you're looking at about 24 samples, and with that P4 flow cell, about 36. And now, of course, if you're focused on exomes, generally we recommend about 8 gigabases or about 100x coverage. This could, of course, vary depending on the application you're focused on, whether if it's clinical, you might want a little bit more of a higher throughput sort of read, just to make sure that you're looking for the correct SNPs, SNVs, CNVs. But for a 100x exome here, you're looking at about 67 exomes on a P4 compared to about 45. So those savings from sequencing can allow you to spend more to focus on expanding your projects further even. If we think about single cell on a P4 flow cell too, just given the amount of output, you're going to be seeing a pretty large increase in the amount of cells that you can fit per flow cell here too. Additionally, generally we might recommend about 20 to 25,000 reads per cell in these cases here. You could even go a little bit further with the amount of sequencing you want to do, whether maybe if you're thinking about a 10x run where you can run different sort of multi-omic kind of modalities added to it, you can increase the amount of sequencing depth needed to get a little bit more output on each of these kind of higher throughput P4 flow cells too. And additionally, as I just called out, this could also just result if you're keeping it at 20 or 25, an increase in the amount of samples that you run to about 18 here on a P4 flow cell when you're thinking about keeping it to about 20,000 reads per cell here. And I also just want to call it that we have internal data too to show that we've tested some of the single cell sequencing data internally on our NextSeq2000. So we've used the P3 100 cycle kit, which is recommended for a 10x sequencing run. And we're comparing it to a XLEAP P3 100 cycle kit and P4 100 cycle kit. And you can see that we're mapping quite well with our UMIs, our valid barcodes, and the reads mapped to the genome. and the reads map confidently. You can see that we've actually exceed in a few of these places here across the board for both P3 and P4. So now when we think about spatial sequencing, and depending on what your focus is, whether it's Visium HD, you can start here. You're looking at, you go a little bit of wiggle room for scaling up your the amount of samples per flow cell you might want to work with. So maybe you are focused on Visium FFPE. That'd be about 3 samples on a P2 flow cell. But you can also scale all the way up to about 14 samples on that P4 flow cell. So a lot of room to grow with your scalability here. And as Visium HD, which is 10x's new sort of spatial application, starts to roll out, that requires quite a bit more sequencing depth. And that's kind of nicely where the P4 flow cell sits already here, which you can utilize for getting the kind of read depth that you need to get as much unique transcripts from this specific application. And then maybe perhaps you're interested in nanostring, or that might be your spatial platform of interest. For your regions of interest, we have, once again, a few different applications that make the most sense. Probably a P1 isn't necessarily a perfect fit unless there's one specific region of interest you're interested in, but you can go all the way up to about 18 regions of interest sequenced here, about 100 million reads per sample in this case. And then when we think about, well, in this slide, I'm not going to go through every single one. I'm happy to share these slides afterwards. But as you think about applications outside of RNA-seq, and that's the question I usually get the most, which is how many samples per run can I get onto this flow cell here? This is just kind of a loose graph, and depending on your application, I'm always happy to answer questions to give you a little bit more information and what makes the most sense based on literature out there, or kind of what your focus of your experiment is. But we can share this afterwards. I also see there might be a picture being taken, so I'll wait 1 second too. You're all good there. So any sort of application, we're here to work with you to find out how to run it as optimally as possible. So turning now to the NovaSeq X, different flow cell configurations as you scale even further than that 1.8 billion reads. There are 3 different flow cell configurations on the NovaSeq X: the 1.5 billion read flow cell, the 10B flow cell, or you're getting 10 billion reads per flow cell there, and the 25B or 25B flow cell. So names in this case are a little bit more self-explanatory, and how many billions of reads that you're getting here. Sort of, I do want to call it, you're actually getting 1.6 billion reads, not 1.5. 1.5 rolls off the tongue a little bit better. And also you're getting about 26 billion reads, not 25. Once again, I think it just probably rolls off the tongue a little bit better in these cases here. But you're seeing quite a few different flow cell configurations. You have your 100 cycle to 300 cycle kit and the 1.5B. Currently, right now, we only have the 300 cycle kit for the 25B on market, but we are going to be coming up with a 100 cycle kit, which will be beneficial for single cell sequencing specifically. And additionally, you have more lanes on these flow cells. So 1.5B, you have 2 lanes. So you could theoretically do 800 million reads per lane, depending on the size of your project there, if you need to go smaller. But you also have 8 lanes on the 10B and 25B. So you don't necessarily need to max out this flow cell to kind of reap the benefits of it. Maybe you only need 2 lanes, maybe 3 lanes, things along those lines. You can still run different library types on each of these flow cells if they're running in different lanes here. And then we're all once again calling out the Q30 scores that we're seeing here too. So putting that into scale once again, how many samples can I fit onto each of these flow cells? What makes the most sense? This is comparing the S1 and the S4 flow cells from the 6000. I should also call out though the S1 is most comparable to that 1.5B that I've shown. So these calculations here still compare and make sense. And the S4 compares to a 10B flow cell too. So if you're thinking about whole genome sequencing, as I called out before, you have your 24 samples on the S4 or 10B. You're getting up to 64 whole genomes here in these sort of cases. For whole exome sequencing, you go from 250 samples on an S4 to 750 samples. For whole transcriptome sequencing, 200 samples to about 520. And then for single cell sequencing, 25,000 reads per cell can go from 400,000 cells to 1 million cells here. So a lot of room for scalability with the NovaSeq X as you maybe start with the 2000 and move over to the NovaSeq X based on the kind of parameters of your experiment there. So just kind of give an idea what some people are already using the NovaSeq X for, kind of where this can scale quite nicely. We have a few different papers here. We're not going to get into the weeds on every single one, but just putting in scalability of how they're using the potential of the NovaSeq X for their experiments here, or how they could possibly have used this for their experiments here to get the most cost savings. On the far left, the 1.5B, that's once again a great application for gene expression profiling. getting about 32 samples in 13 hours with using 50 million reads per library. Additionally, if you're focused maybe on cancer and you're doing a mutational profiling of a tumor sample, you're looking at about 62 samples if you're getting 160 million reads, because you need to go a little bit deeper in these cases to find that needle in the haystack, perhaps. And then, of course, maybe you are looking for that biomarker, that cell-free RNA library or kind of a fragment in a liquid biopsy sample to try to find maybe something that'll let you know if a genetic disorder is oncoming here. There's been a lot of interest in liquid biopsy with the announcement of the 25B, just because you can get a little bit deeper. We're seeing interests in liquid biopsy for cell-free RNA, for proteomics, for methylation. If you're interested in any of those sort of applications and how you can utilize the NovaSeq X for these readouts, always happy to have those conversations too. So that was RNA-seq specifically. Turning now over to single-cell sequencing. If you're focused now for maybe doing multiple samples and treatments at one time to see how a cell line might react to the treatment that you're working on, you can look at about 120K or 120 single cell libraries per 1.5B flow cell. This is assuming a dual flow cell run. So you're loading two 1.5B flow cells or 60K per 1.5B. Maybe you want to do a larger kind of atlas study. You're trying to build an atlas of a larger complex tissue type, as done in this paper here too. You have the 10B flow cell for this sort of application where you can get really deep. And then for those who might not be familiar with Perturb-seq, this is a fairly new paper that came out from the West Coast here, where if you're interested in maybe CRISPR knockouts, as opposed to doing a CRISPR knockout for maybe a specific mouse model or one individual cell line at a time, by using single cell sequencing and kind of a guided approach here, you're able to take cell lines and do single cell sequencing and look at how the genotype affects the phenotype across the board with this sort of approach. So instead of spending a lot of time doing all these different knockouts, you can do them all in one go on the NovaSeq X, getting about 1 million reads or 1 million cells per 25B or 25 million in 48 hours if you're using both sides of the instrument there. So I know that was a lot of information. We just really wanted to showcase that the NovaSeq X and the NextSeq2000 have had a lot of work put into them for using XLEAP chemistry. As you think about your experiments, we have a lot of guides for helping you along the way if you don't want to reach out to us right away necessarily. If you're focused on cancer research, liquid biopsy methods, multi-omics, so genomics, transcriptomics, epigenetics, proteomics, and other single cell methods, we have guides for kind of what's on market, either with Illumina or not with Illumina, what we're seeing and what you can do to really expand your research. So with that, I just want to say thank you guys very much for giving me some time to come up here and talk about some of the XLEAP chemistry. Chevelle will be coming up in a second here to talk a little bit more about what you guys should be aware of when using this XLEAP chemistry on the NextSeq2000. So thank you guys. And if there are any questions, I think we'll probably save them for the end. So does that sound good?
Yeah. Hello, everybody. My name is Chevelle Blackburn. I am the primary field application scientist for you all. What that means is that I work with an entire team full of field application scientists that have different backgrounds, and depending on what issue might come up, we are available for certain types of troubleshooting and all types of training. So if you were to have questions about a particular application, adopt a particular application in your lab, or let's say you were just trying to figure out what was going on with some data, or figuring out a new application of sequencing that you wanted to try out, I would be your typical point person, and I am local to the Chicago area, so I also come on site depending on what the issues are. For today, I'm going to be giving you all a general introduction to the P4 flow cells and XLEAP chemistry on the NextSeq2000 system, and then I'll just be talking a little bit about some differences if you've used the standard kits versus the XLEAP ones. So the consumables look exactly the same. You'll still be getting the reagent box that also has the cartridge in that foil bag. So we've got a reagent cartridge that has pretty much everything that you need in it, and a flow cell that comes in its own individual bag. We prepare those and load those onto the instrument. We don't really need to look at the recycling tool. So why you all are here and what we're most excited about is this new release of the P4 flow cell. What that means is that we have our standard methods of how you can sequence on the NextSeq 1000/2000, and we have a newer version that's currently available for the 2000 instrument, which is what we consider our high throughput. The 1000 is kind of the mid throughput option. And so there are a couple different kinds of flow cells that you can use on that 1000, you can use all of them on the 2000. And then, like Logan mentioned, we have a couple new flow cells that are going to be coming out later in this year that you will be able to use that have that XLEAP chemistry on both the 1000 and 2000 instruments. So, like I said, the P4 has been released, and I think we're up to like 1.8 billion reads. So, we're seeing stellar performance on the kits that are being released, and we've been using this XLEAP chemistry on the NovaSeq X, so we feel pretty confident about it as we're releasing the P1, P2, and P3 kits. You'll still be able to use both the standard and XLEAP chemistry, but in order to use that XLEAP, you will need to upgrade the instrument so that it is on the 1.7 version of software.
Do you know if there'll be an update when the P1, P2, P3 come out?
I do not know.
Anyway.
Yes. I was just reading over some things earlier and I didn't see anything that called it out, but I'll look into it just in case. For the P4 flow cell, it will come in a different color, so it will look distinct and different from the previous standard chemistry. And there's a 50 cycle, 100 cycle, 200 cycle, and 300 cycle option for kit output, depending on the kind of sequencing that you are planning on running. Like I said, we've got color coding so that we don't confuse these. You aren't able to interchange the flow cells with the kits, so they do need to be matched up with what type of kit you're using. And you will want to decide how much information you would like to see from your instrument to decide what version of that flow cell you're going to be using. There are benefits to using both, if we're just being honest, but the major difference between the different flow cell types outside of this particular chemistry is the width of the lanes on that flow cell. So it's kind of like the real estate that you have available to you as you're generating clusters in your sequencing runs. Oh, I feel like this might be in the way. And what is this mouse? Okay. No, it's all good. So I just wanted to show you all some size differences. So to be clear, the XLEAP kit is no different in size than the 600 cycle cartridge for standard chemistry. What that means is that it's just a little bit longer than our regular like 300 cycle or less kits. But you don't have to make any changes to the instrument. It is equipped to deal with the size change from that 300 to 600 kit. And so the XLEAP kits can be loaded onto the instrument without any additional changes. There we go. Let's talk a little bit about the workflow, just so that you all know what differences need to be called out and in the event that you decide to use the P4. So the typical workflow is that you thaw your cartridge, you'll prepare your libraries, you'll load your libraries into the cartridge, and you'll start the run. The process for thawing does look a little bit different. I'm just gonna drag that over. You want me to just get rid of that? I'm gonna get rid of it. We're not even gonna worry about that. I've got like a summary chart so that you all know how long you can thaw depending on the method of thawing that you're using. If you're using a controlled water bath, that's gonna need to be at 25 degrees Celsius. And we've got an 8-hour window, which is a little bit longer than our previous standard kits. And then for the refrigerated method, which is the more commonly used one, you would leave it out at room temperature for 6 hours, making sure that there is airflow on the sides and top, so not stacking things on the side and on top of the cartridge. And we are thawing in the foil bag. So you'll take that cartridge in the foil bag and put it into the refrigerator now for 16 hours, which is a little bit longer than the standard chemistry. Room temperature is also a little bit longer, but we have charts so that you all can see the P4 kits versus the P1, P2, and P3 standard kits so that we don't confuse those thaw times. We're still looking at an overall 72-hour window of use. So in the event that you start thawing a kit and you're not going to use it that same day, you can store it in the refrigerator depending on the method that you start it with, and you'll have a window of time that is available for you to continue using that. Just make sure that you don't refreeze the cartridge in the event that you start thawing it and realize that you're not going to use it. Refreezing will mean that the enzymes might not work as well as we want them to, and then they won't be supported. So in the event that something does look wonky with your run, we won't be able to replace it against the consumable warranty. Okay. So we have updated our guides that are on our support page. So when you go in to prepare your libraries and you're trying to figure out what are the recommended starting loading concentrations for how we're going to put your library into that cartridge, you'll need to go in and determine, are we using XLEAP chemistry? And then there's also like a separate section that will talk about the standard chemistry so that you all know how to appropriately prepare and load up those cartridges. The loading concentration may also vary by the flow cell type that is used, so just keep that in mind. Those differences will be called out in the guide. There is also a difference in the two-channel chemistry that's being used now. So we're still using blue-green chemistry, but now the standard two-channel chemistry for the standard kits on the NextSeq2000 are that the combined like blue and green signal represents the A. Now for the XLEAP chemistry, the combined is the C. So the biggest thing to call out here is just making sure that your cycle 1 has some signal that originates from an A or a C. This particularly comes into play if you're using like an Illumina RNA library prep, where like 100% of the T's that are at the beginning of the sequence could then kind of result in a failed run because it's not seeing signal in both of those. So just one of those things where we need to keep color balancing in mind. But in this case, we would just recommend that you all use like at least 5% Phi-X when you're spiking in, if you know that there's going to be a bias for that first cycle when you're sequencing. When we're going to load our libraries, typically the process is that you'll thaw the cartridge, which can take a day or two, and then you can prepare your libraries. So just before you go in and pierce that foil seal and load the libraries in, you will want to invert that cartridge slowly like we were before. The difference is that with XLEAP chemistry, we do need you all to kind of do a couple taps that will help release some of the bubbles that can form in that inversion process. So we saw in internal testing that without doing these taps, which is basically like lifting the cartridge from the arms and dropping it on the bench about 5 times. If we don't do this process, sometimes that can end up in wonky data when we're looking at the final output. So this is now added into the workflow just for the XLEAP chemistry. It is going to be the same in that when you move it around, you will hear that rattling piece that does all the piercing of the foils inside. So we don't need to worry about that as long as we're following the proper thawing procedure, but just make sure that you do that lift and drop so that we can move forward and start your run. Here's just a summary of our P1 through P4 kit specifications. Each one of our Illumina kits have specifications for what we would consider good or passing. So this just summarizes kind of what we're looking at if we're trying to see the overall quality or the goodness of a run, as well as how many reads we should be expecting to see, the output depending on the type of library and the parameters of your run, as well as the runtime. So this can be a helpful reference if you're trying to figure out, is this good, is this not, how do we troubleshoot, and is this something that needs to be replaced? Logan kind of went over this in detail, so I won't belabor it, but just know that we are seeing very positive data in terms of the error rate being lower on our XLEAP versus our SBS chemistry, our standard SBS chemistry kits. So we're feeling very good about that. And then we are seeing a significant increase in Q40 scores. So that just means that our base call accuracy is even higher using the XLEAP chemistry.
So there are a few different companies out there right now that offer different kind of technologies for using Illumina sequencers to get a proteomic readout. They use kind of NGS as that readout, and they develop their pipelines to say, hey, in this liquid biopsy sample, for example, we're seeing this concentration of proteins compared to this concentration of proteins. And because of, and this comes down once again to pitch of these flow cells here, you're really able to get quite a large dynamic range all the way from 4 log to 10 log pretty much here on the how deep you need to go. So when you think about Olink, they have the ability to break down some of their flow cell or some of their panels. So if there's a specific area, maybe immunology that you're focused on, you could use a NextSeq2000 for an individual panel, as opposed to maybe doing a wide proteomics study. You can get about 5,000 unique proteins and run that on the NovaSeq 6000 or X. Illumina will be coming out with a proteomic panel or a proteomic application later this year. It's currently in early access. But if you're interested in receiving about 9,000 unique proteins, you can get that readout. And also we have visualization tools to show you how that data can be merged with transcriptomic or epigenetic data too, in the form of Partec flow, if you guys have maybe used that for some visualization of data purposes too. There's also a few other companies that use our technology or use our sequencers for the proteomic readout, but it's coming soon. And if you have questions about proteomics, happy to talk about that too.